I will just briefly introduce Tudor, our speaker from today. He's a protein engineer at Adaptyv Bio in Lausanne, where he works on developing and applying technologies for the next generation protein engineering. He completed a Master of Science in Biotechnology at ETH Zurich, and before he did a Bachelor of Science in Biological Sciences from Imperial College London. And during his studies, Tudor gained research experience across several leading institutions, including Max Planck Institute of Biochemistry, Imperial College London, working on topics related to protein design, structural bioinformatics, and synthetic biology. And his work focuses on bridging computational protein design and experimental implementation, helping translate advances in protein engineering into practical tools and applications. And today he's going to introduce Adaptyv Bio, a very successful company, and that's becoming very famous also because of their competition. And I will not say anything more. I will let you speak. The floor is yours, Tudor. Thank you for agreeing to come into.
Yeah. Hi everyone. I'm Tudor. I'm a protein engineer at Adaptyv, and thank you for the invitation for this event. I will briefly talk about what Adaptyv does, and then I will be talking about some cool recent stuff we've been launching and releasing. A lot of it will be about these protein design competitions, as it seems a lot of people really enjoy. But we've also released a couple of new other interesting products, so I'll be talking about that as well. So I'll start with Adaptyv. So Adaptyv was founded in 2021 in Lausanne by two EPFL master students, Julian and Daniel, and currently we're at 25 people. And just in the last year, we scaled by like more than 12 people. So we're growing quite fast and we're scaling quite a lot. And the main thesis of Adaptyv, what we're basing this entire company on, is that protein designers should be designing, not pipetting. So in a nutshell, we deal with the experimental validation, the experimental bottleneck in testing your protein designs. So how Adaptyv works, it's a pretty intuitive, quite simple platform to use. We offer a set of assays. Currently, we offer binding affinity assays. We also offer chromostability, expression, and in the future, we'll expand to some enzyme activity assays. And you choose your data type, your assay. Then in our Foundry platform, you can upload your CSV of protein design sequences. And that's all you have to do. Then we'll run the experiment for you. And in 2 weeks, you'll get all of these nicely presented results. And you can use that for downstream analysis. You can use that to train a model. You can use that in like some agent in the loop kind of workflows that we've seen being more popular recently. So we operate a fully automated lab, a lot of hands-off kind of work from our wet lab scientists, and all of our assays that we offer now are fully automated, and we're also vertically integrated. So we do the DNA synthesis into expression, into binding assays, and then the QC and data processing. And everything is done by us. So in the end, you just get this clean experimental data set. Like for example, for binding, you get like really nice curve fittings, you get KD values, and everything is well explained. And recently, we also made our API public, and that means is that you can do programmatic experimental testing. So in the same script, you can generate your designs and then call this API endpoint for us, and then that submits your designs to the lab, and then you can like query that to get your results, and everything just in a single Python script. So we're also powering the world's best protein design teams with this infrastructure. We've tested more than 100 targets, 10,000 proteins expressed, and 100,000 binding curves generated, and we're scaling that more and more. So the main things that Adaptyv is doing is we want to unblock the design test bottleneck. Currently, that's the main limiting factor in actually either improving your AI generative model for protein design, or even like taking a molecular therapeutic and getting it further into the therapeutic pipeline. And with this, we by unblocking this design test bottleneck, you can do like faster design, build, test, learn cycles. So you can have a model, generate some design, see what works, what doesn't work, learn that, or like fine-tune your model, retrain your model, and then have a better model to get even better proteins in the end. And ultimately, we run this lab, so you don't have to deal with any setting up your own lab or like contacting CROs, CDMOs, or having to like send Excel sheets or CSVs to someone and then begging them for like 2 months to get experimental results. Everything is super streamlined into our platform. So the main thing, like what users see, is this Foundry portal we like to call. And then here, pretty much you can set up your experiments. You can see like how far your experiment has progressed, like if we're waiting for materials, if it's in queue, if it's already done, if it's like in post processing and QC. So everything is fully visible there. And in the end, you'll see, for example, for binding affinity, like these really nicely formatted bar plots. And then you can also download the entire data package. And for binding affinity measurements, that includes the KDs, the K-ons, K-off values, and also the raw sensorgrams that we're providing, so you can do your own fitting and see. So ultimately, we have a sort of stack built on top of this. So we have the user portal, like the front-facing user interface, the Foundry portal, and here you can manage experiments, upload proteins, and also get the results, get the data package. And internally, we manage all of these experiments with what we call the lab OS, sort of our brain behind the lab. And this both orchestrates the automation and does like inventory tracking, material tracking, plate tracking. We do like the workflow management, so everything is fully visible, so we know like at which step we are through the workflow. The processing, curve fitting is also conducted through this brain, through the lab OS. And this pretty much operates our entire lab. So when you think about us, we're ultimately kind of like a liquid handling company with a lot of nice stacks on top of that. So everything is done by these liquid handlers into these gators and Cartera machines for either BLI or SPR binding affinity measurements. And again, this is automated, so we have work cells running through this, like taking plate by plate from the expression, putting it into the gator, running the assay, and then we have the automated QC pipeline at the end for curve fitting and seeing if these results are robust. Currently, we offer a set of assays, but we're also quite rapidly expanding. So we do a lot of cell-free expression at a sort of basis, and from this we build the entire assay stack. And we offer binding characterization, epitope binning, permissibility. We can also look into like non-specific binding or other modalities for binding. We're building some enzyme activity assays that we're bundle up and will release quite soon. And we also do like some sort of like developability package with some aggregation quantification as well. But we also want to further expand this and streamline even more assays so people can have access to that and stuff that's even more important downstream in the therapeutic pipeline. So like cell activation assays, immunogenicity, and also we want to scale up our production even more, our cell-free synthesis, so then we can also offer you these recombinant, these expressed proteins if you want to like use them in your lab and you don't have the infrastructure to express them. So this is sort of a mini bragging slide, like the teams we've been working with. We worked with Boltz Bio. We validated their entire Boltz Gen model for like these 12 difficult targets from PDB with minimal overlap to the training data. And we're pretty happy that we delivered these results quite fast end to end. It was quite a large experiment. For Chai, we also validated their Chai-2 model, and they did also like quite a wide array of targets that you can see binders from this model. And in this one, we had a 2-week per round results return rate. And then we're also working some pharmas as well and some other frontier AI labs. I would say now we're working a lot with these AI labs that are tapping more and more into protein design. They have like the machine learning expertise, but they don't have the sort of desire to build their own lab infrastructure. And they've tried to work with CROs so that it doesn't really work that well. So they prefer to work with us simply because our process is more streamlined and more intuitive. But we're also working with academic labs, other like smaller industries or startups that are just getting started and they want some initial validation for their models, either for like pitching, to like raise a series or anything like that. In this sense, we kind of describe ourselves as the layer, like the infrastructure layer that enables the future virtual biotechs. So people that ultimately would only use computational resources, tools, and have like the domain knowledge to develop some therapeutics, but they don't have the experimental infrastructure to actually do all of that testing. So we're like sort of the first step in that direction. And in 2025, we also scaled our lab output by more than 5x. So we tested more than 20,000 actually unique proteins with 200 unique targets tested just in 2025. And you can see like this just going up and up and up, and it's going even like more up as we're speaking. So our main sort of our bread and butter assay is this binding affinity. We're doing both BLI and also SPR, and we use Carteras, we use Gators for this, and pretty much it starts from you sending us the protein designs. We do some DNA optimization, some codon optimization for this. Then we do DNA synthesis. We have been working with this, but recently we also verticalized and we're doing more and more of in-house gene synthesis. Then we're doing software expression from this, and then feeding everything into these machines to get these quantitative results for KDs. And then we have a pipeline for the post-processing, curve fitting, and showing you the results in our UI. So we have been working with quite a large target catalog, and you can see that in our Foundry portal. We have been quite successful with a lot of targets. Some targets are just inherently trickier, but we're pretty happy. We've been so far successful with more than 90% of our targets. And it's pretty easy for you in our Foundry portal to look for our target catalog, see any of the targets we have, see if we've worked with them, get some like more information like the species, the region that we're having, the tags for that target. And then you can just say, okay, I want this target. And then further down the line, you input your sequences and then we run the assay for you. We do both target expression in-house, or sometimes we also source targets from Acro and Sino if we want like a higher scale expression. So this is the sort of the actual experimental setting, what we're doing experimentally, but a couple months ago, we launched this thing called Proteinbase, this web page that we call it sort of like the GitHub/Hugging Face of protein design. So the main idea was that we had a lot of internal data, we had data from these binder design competitions, and we were like sharing that via GitHub, like uploading a CSV, and we weren't really happy with that. We thought, okay, we have a lot of data we can open source, we can share that with people so that they can train better model, or they can look into our data, either like pick a protein from there and use it further down the line. And we wanted a nice, more intuitive way to showcase this data. And that's why we built Proteinbase, the sort of the home of protein design data. So in here, you can see like this sort of concept of a collection, which is just like a set of proteins, either with experimental validation or without, that have like some nice structure predictions and other metrics we're predicting from our internal pipelines. And then you can also run these through the lab and you see, okay, this was a binder, like this was experimental validated or it wasn't. And you can easily sort of save these, you can upload these, you can share these with people. And also it's a nice way for us to host our competition. So everything is done through Proteinbase. People participate, they create a new collection for this competition, and after that we do the validation and they also see who won or who ranked best also on Proteinbase. So yeah, this is also another, like the main sort of collection page. For example, this from the BoltzGen release, we open sourced the experimental validation we did for BoltzGen on Proteinbase. And here you can see like all the targets they designed binders against, the sort of best binder, the best KD, how many binders they have, and just some overall metrics like how many proteins in total, proteins designed, validated, how many binders, the expression rate. And I'm also really happy with how these structure like graphics look. Now, so as I described earlier, we're hosting our protein design competitions on Proteinbase as well. And I want to talk about one of the main competitions, like the one that started all of them. It was this EGFR binder design competition. So we tasked people to design new binders or to improve existing binders against this EGF, epidermal growth factor receptor, that's also involved in cancers. So there are some guidelines. For example, have 3 weeks to design the proteins, at the most 10 submissions per team. We didn't want people to just copy paste rituximab, for example. So we also asked them, okay, they have to be at least 10 amino acids away from known proteins. And in the end, we validated 400 experimentally for this round 2. And the outcomes, we had 130 protein designs participating, 1,100 novel designs submitted, pretty high expression rate, and quite a lot of binders that we saw in the end. And we were really happy because we saw people taking different approaches, using different models, either doing de novo design, so creating a brand new binder, or taking existing binders and modifying them a bit to do lead optimization. And even in the diversity that we saw in the binders, we saw antibodies, we saw nanobodies, these really cool solenoids that some people were designing, or we also saw people, for example, in the second round of this EGFR binder design competition, they took a binder from the first round, added a linker, and then had like a new binder that they made to like leverage some avidity effects for in this round. Ultimately, in the second round, we also saw something quite cool from this other company based in Switzerland called Cradle. They are doing machine learning for protein optimization. And what we saw is that they had a 9x increase in KD versus Merck's cetuximab with the antibody they designed. So that was quite surprising. Also, like, it was a nice validation for their platform as well. And they ran a lot of PR as well after this competition. And we also saw 2 binders that were better than cetuximab, and overall 29 that were better than the natural binder EGF. Here I'm comparing sort of the progression from round 1 to round 2. I didn't mention, but we first hosted like a smaller round of this competition that went quite well, and then we just decided to expand it even more into a round 2. And the cool thing was that people kind of took the information, like the knowledge from the first round, and then applied that to the second round, and then we saw a lot more express design, so from 73% expression rate to 94% expression rate, and then we also saw an increased hit rate from 3% to 13% hit rate in the second round, and that was pretty cool. And then we also looked into like the sort of design choices, the design methods people we use the most, and a lot of people mentioned, oh, I saw in the first round how this Minecraft pipeline did the best, so then I used that, or I used this soluble NPNN redesign tool to solubilize my sequences, and it was just interesting to see sort of like the progress and tracking this progress across the 2 rounds of this competition. We also did some postdoc analyses that we published in this blog post and also a community paper. And we're mainly interested in seeing which sort of in-silico metrics best correlate with experimental outcomes. We just wanted to ask the question, are we there at that point where we can directly predict binding, or we can predict expression purely from computation models? And the main conclusion is that unfortunately we're not there. So looking at these sort of correlation coefficients between some commonly used scores, such as the IPAE from AlphaFold, so IPTM from AlphaFold, and this ESM naturalness likelihood. And there are some papers that show, for example, from the Baker Lab, that some of these correlate more or less with both like discriminating between binders and also with binding affinity. In our case, we were not able to observe that, we ascribe that to we saw a lot more novel designs that are kind of out of distribution. So these models weren't trained on these new sequences we tested. So maybe that's why they didn't perform as well. So ultimately, like the main conclusion is that we still can't sufficiently predict experimental outcomes purely from in silico metrics. So we still are bottlenecked by the experiment. We still need to validate these proteins in the lab. Afterwards, so as I mentioned, we wrote this community paper, and we had like a channel where we discussed with people who participated, with a lot of protein designers, and the most common theme we found was that there was almost no way to standardize or to benchmark protein design, as in some people use these targets, some people did it this way, some people had like these experimental settings, yada yada, and like we saw a lot of people just sort of copying what a previous paper did, but it wasn't necessarily standardized. So then we decided to select this set of 7 targets. We wanted something that's both accessible, people can express it quite well, like the maltose binding protein, but we also wanted something that's kind of like novel, like this BVF protein, this beta barrel, that's de novo designed protein from Bruno Cotet's lab. So this is not in the training set yet for stuff like AlphaFold. And we also have like stuff like CRISPR, some like therapeutically relevant proteins like EGFR, PD-L1, and we also defined the sort of threshold in binding affinity for a hit, and then gave some clues of like, or some tips and tricks for what experimental setting would be best to reliably benchmark your model. So using stuff like, again, BLI, SPR, all of these. So you can find more information about this at benchpb.com. And we also have this preprint, like the community paper we released after the EGFR binder design competition. So we were pretty happy with the EGFR, the 2 rounds of the EGFR binder design competition. So then we launched like a sort of 3rd round. In this competition, we decided to select a new target. We were really interested in this Nipah virus. We saw some headlines like a couple of weeks before launching this of like, oh, there's like a mini outbreak somewhere in India, and we were like, okay, maybe we can do something like either for like pandemic preparedness, maybe people design some interesting antibodies for this, so that's why we chose this as a target. So to give you some numbers, we had 680 protein designers participating, which is way more than we expected for this one. More than 10,000 novel designs submitted, and we validated, actually that's a bit wrong, so in the end we validated 1,200 proteins. To show some results, we also published these on Proteinbase. Everything is open source under an accessible license, so you can use it for commercial purposes as well, or to train your machine learning models. And the main result is that we saw a lot of people getting like really high, really tight binders. These people, for example, work for this company called Instadeep that does lots of machine learning, and they had this model in-house that they wanted to test. That was pretty cool to see. This person is, I think, an undergrad now, where he's still in high school, and he just tried out some models, participated, and had a lot of fun, and also ranked 3rd. And then we also wanted to give like a special prize to the de novo winners, because in most of these cases, these were gate optimization cases. So we wanted to also see what the best de novo protein was. And you can see it's like these helix bundles, for example, from Nick Boyd from this startup called Escalante that developed a pretty good model for designing de novo proteins. For example, in our competition, they had like a 90% hit rate, which was really impressive. And we sort of didn't stop with all of these competitions. We organized many more. We organized a couple of them in like a hackathon format, and a couple of them in like an open accessible format. So the hackathon format means that you just have 24 hours and you need to be present either at EPFL or in San Francisco or in Berlin. But this one we co-organized with the ICLR, with this machine learning conference. They have this workshop called GemBio that looks at the intersection of machine learning and experimental validation. And we co-organized a competition with them. people pretty much just submitted a sort of mini paper. They described their method in 2 pages. Then they sent us a set of designs, and currently we're validating these ones, and we will be releasing the results in 2 weeks in Brazil, in Rio de Janeiro. So we're pretty excited about that. And as another thing, what we've also been building is this API that we launched quite recently. So this means that we want to make protein design more programmatic. So we don't want people to even have to click through our Foundry portal to like get to a page and then to see, okay, I have to like submit my CSV or my Excel sheet here, and then I have to do a bit more clicking. Like we want everything to be super automatic, easy to use, so you can easily just input this into your protein design script that calls like a model such as OldGen, and then it does some selection, and then it calls this API endpoint, and it submits the designs to us directly. And this works quite well also with these agentic workflows. So we've seen more and more, for example, stuff like Code, tools that have this model context protocol that ultimately calls them an API as a tool, and then the chatbot, the agent, knows exactly what to do. So you task it, okay, design some proteins for me, and then submit these to the Adaptyv Foundry, and it does that exactly for you, because in the in its backend, it just calls this API that we made for it. So we want to enable more of these sort of autonomous discovery workflows, in which you just launch code, you task it with design, fetch PDL1, design some binaries against it, submit the designs to Adaptyv. Once you get the results, do some data analysis, and then select the best protein with the best binder, and you can easily just fully automate that process through the API we built. So yeah, I pretty much showcase like some the platform, what we've been working on for this wet lab validation, the infrastructure, and also some cool things we've been doing, such as the protein design competitions, Proteinbase for open source data sharing, and also the API we launched recently. And now, we still have some time, so I wanted to actually showcase the Foundry portal. So what the clients see when they have access to our portal, and how they can submit designs. So I will stop sharing this, and I will share this one. Can you see this?
Yeah.
Okay, so we're gonna, okay. So this is a Foundry portal. This is what our customers have access to. They have like this organization, and because it's Adaptyv, they can invite members. Here they can also get API tokens for the newly released Adaptyv API. So you just get like this code, and then you can call our endpoints to programmatically submit and retrieve results. And you can do like some billing. You also have the documentation here. So we have these sort of experiments, like these are the main concepts, like some competitions or like some random tests we did here. And the main workflow is that you can click to create a new experiment, you can name your experiment, you can select sort of the assay type that we're offering now. And again, we're expanding this. The cool part is that the pricing is pretty accessible, so you can say, okay, it's 99 per design, and then it automatically calculates the price for the whole experiment. We also have some discounts if you want to open source your data on Proteinbase. We have like 20% discount. So then you can like go for target catalog, and you can check all the targets, or you can add your own custom target, like some acid sequence, Uniprot ID, if you source it from a supplier, and any other information that we need to know. So you just go here, you have some targets, you select the target. This is sourced from a supplier, so you can see the supplier page. You can get more information about it or something from Uniprot. And then you just upload your design. So you can also, we support multiple formats. We support just single-chain proteins. We also do scFvs and Fabs. We're building towards IgGs as well in the near future, so we'll be also offering that as a new format. So you select your format, you drop your CSV here, and then you select like the number of replicates, and you can create a new experiment. So this is the experiment creation workflow. And then I wanted to also show you sort of some results, like what the results page looks like. Let me find something from our competitions. We can look at this EGFR round 2. So we can see it was like an affinity characterization when it ended, status, some description of like the sequences, average length. We can also see like the positive controls, different sequences, like their names, or was like the participants in the protein design competition, how they named it. You can also download this data package. It pretty much gives you a CSV with the per replicate results, K on, K off, and you also get the raw data, such as the sensorgrams we get from both the gators and the carteras. And then you have like this nice bar plot where you can just see like your best binders. You can also rank over here by affinity, and you can see, okay, this is like the cradle binder I showed earlier. You can see like the EGF control, how well it positioned, and you can see like all of the binders from everyone, and some that were non-binders as well. And also you can see the sort of expression, like the success rate. We have like these expression labels like high expression, cell-free, medium, or low. Here everything expressed because we were able to validate it. And yeah, we should also be able to compare to the control. For example, in this case, we have the EGF, the natural binder, and you can see which ones are better than this and which ones are worse. So you have like all of this in the data package. You have like some labels for binding strength we add. It's just stuff like if it's below like if it's between like 1 to 5 nanomolars, it's strong, or like higher than that, we add medium, or if it's like between 5 to 10, it's like a weak to non-binder. So you get all these labels, you get the KDs, you get the K-on scale-offs, sequences, like all the information you need. So this is pretty much like our main portal, our main like what the customer sees when they get access to our platform. So for them, it's really easy to just create an experiment and then submit that, forget about it, and then in 2 weeks they get a data package with the results, and they can further use this in downstream workflows, analyses, or just to train new models. They can download the data package, and now they also have access to this API where you can create a new token that you can use with our endpoints. Yeah, so this is the sort of platform showcase. Hopefully it's simple and intuitive. We really wanted it to be this way, so people can have like a really easy time just designing proteins, submitting them to us for experimental validation, and then they don't have to worry about anything else. Yeah, so this was my presentation. Thank you.
Thank you so much. Super interesting presentation, very impressive results, both from Adaptyv, but also from some of the designs that were submitted to your competition. And I love the commitment for making everything, the data and the process more easily accessible. It's really cool. As usual for the audience, you can ask questions both raising your hand and unmuting yourself, or writing them in the chat, and I can read them out loud for you. So during your presentation, some people have asked some questions already in the chat, so we'll read them and you can answer to them, or you can read them directly if you prefer. I can read them out loud still.
Yeah. I can go one by one. So someone asked, how do you find workarounds for producing difficult to express proteins? So this is a bit tricky in a sense. Like we do E. coli cell-free as our main expression system. We had some pretty decent results with that. Especially when you do like DNA optimization, we get some pretty decent results, but there are other proteins that are just hard to express. For example, like in my master thesis, I worked with these enzymes, like these enzymes that are involved in PET catalysis and plastic breakdown, and these are almost impossible to express in E. coli cell-free. So ultimately, we tried other expression systems. We are also working on optimizing our internal expression system. I'm not sure if you saw the Ginkgo Bioworks paper that also did something like that. So we're also building a better expression system for ourselves. We also use some yeast expression systems, this one called Alice cell-free expression. So we're just testing and benchmarking a bunch of them. We have some really hard proteins to express, and ultimately, if one of these systems works best for these, for one of these harder proteins, we'll use that. But in most cases, we do E. coli cell-free with something like PureFrex, that's our main one, and that works quite reliably. Was protein purified from cell-free expression? We do a lot of crude quantification actually, because that's the fastest. So you can ask us, hey, I want this to actually be purified before doing BLI, and we can do that. And if you're afraid of like any off-target or anything like that, but we're pretty open about the fact that we often do crude quantification. For example, for expression, we have like this GFP sort of system that works real well with crude. So that's just faster. Like our main goal is to lower the latency for you to get experimental results. So if we can do something in like super fast, super reliable that gets you there, then we're going to do that. But if you have any concerns and you ask us, okay, I actually want to do purification, we'll also do the purification for you. And also in these like BLI or SPR in these machines, you'll often have like tags, like strep tags to your sensor or something like that. So that's kind of like a mini purification when you think about it, when you wash your protein over that. Alkaline your protein expression systems. Also in vivo, how much protein we get per cell reaction. I'm not necessarily familiar with how much exactly we get, but for diversifying, again, we opt for something that just lowers the latency. So that's why we're not doing that much in vivo, that much like E. coli recombinant kind of expression. That's why we prefer cell-free. It's just the fastest way to get experimental results. Yeah. So the biggest experimental bottleneck or data missing right now is fully unlock the potential of protein engineering. Anything that moves from in vitro, I would say. So we're doing a lot of in vitro. I think we might reach a plateau for in vitro assays, especially for binding. Like binding doesn't tell the full picture, especially for therapeutics. That's why we're also moving to expanding our assay portfolio. So especially for like these therapeutics, we started by building more developability stuff like HPLC, anything for aggregation. But we also want to do more that's not just these in vitro assays, either doing like cell-based assays or like organoid-based assays, like growing some organoids and then testing this compound, and then doing like some simple quantitative things like cell counting, or maybe even doing like single-cell RNA-seq on these organoids to see what actually happens. So anything that sort of unlocks this bottleneck. We're also interested quite a lot in immunogenicity and the assays that we can do for that. So it's something we're researching and doing quite a lot of R&D on already. So anything that moves us a bit further from in vitro, we're working on that. Also, like we're also working on like other sort of in vitro assays. When you don't think about therapeutics, you can think about like sort of industrial enzymes. Like it's another important application for protein engineering, and in that case, we're doing a lot of enzyme assays and bundling those up together. For like petases and petases, we have a lot planned for that as well. Is there a playbook for building out the competition participation for enzyme community? Whatever is the older participant. Yeah, for sure. I mean, ultimately, our competitions are accessible to anyone. For example, the Nipah virus one, the GFR one, you just had to make an account on Proteinbase and you can register and participate directly. That's why we had high school students participating in these competitions. So we're not restricting it to sort of academia or industry or anyone like that, to sort of help like broaden the community and sort of lower the barrier of entry. We also want to like make protein design more accessible. So that means having like tutorials, guidelines, blog posts for people on how to get started, how to design their first protein, how to participate in these competitions. So this is something that we're actively working on. But yeah, is there a playbook for competition participation? I would say the playbook is just be flexible and respond to the feedback. So we didn't assume that we did it right in the first place. So like even people, when we did the first round, we did this ranking based selection based on this IPA metric per mouthful. People weren't very happy with that, and then we just opened a poll online, asked people, okay, what metric do you want to see, and we're just open to that.
Thank you so much. You answered all the questions from the chat.
Yeah.
I have a few questions myself, and also for the rest of the audience. If you want, you can keep asking questions for a few more minutes. My question is, you're running tons of experiments across customer designs, right? So you have a huge data set on the results. Do you think this could help Adaptyv improve even further the current designs, the current models for protein design? And how do you manage the ownership of the data that you produce?
Yeah, so for the ownership, it's a sort of per customer agreement. So the customer says, okay, we want like a special licensing for this, or we want like a special, it's non-commercial, you can't use this for anything. So then the customer tells us what sort of agreement they want for this data, and then we sign up that agreement. So it's like the customer has the agency to ask us for what sort of agreement they want. And for model training, like for models that design better proteins, we don't really want to do that because our customers are already doing that. So we're just like battling against them. It doesn't really make sense. But there are a lot of cases in which we sort of like build an internal like knowledge database to see, okay, these proteins didn't really express in this case, like for this customer, and then we see something a bit similar, and then we troubleshoot that. So it's something that just makes our assays more robust and reliable. So just like faster troubleshooting, faster sort of debugging the experimental results, we're already doing that.
Nice. Another question would be on the fact that, like, in the future of Adaptyv, because from what you've shown, it seems that you're investing not just in single protein specific design validation, but also in this high throughput experimental validation. So, and you've shown that you also have a lot of requests, that these are increasing a lot. So what's Adaptyv's plan for scaling to match these high throughput requests?
Yeah, I mean, ultimately, we believe that we're just like scaling the machinery, that like this is the way we're scaling. So we're just getting more machines. We already have like the sort of liquid handling and automation knowledge to easily build a new work cell. So we just like to respond to the demand, we just build we buy a new Cotet. To respond to these demands, we buy a new gator. I think now we have, it's either us and Roche or Novartis has the most Cotetas. So we're just scaling exactly like this, because we already have like the sort of both the knowledge and the infrastructure to rapidly have set these up and have them online, and then we can already start running some assays on these machines. But there's another sort of way we're scaling when you think about it, and that's like being more vertical. So this is what we've been pushing more and more this recent year, and that means eliminating one of our major bottlenecks in the entire workflow, DNA synthesis. So we already started doing that in-house. So either we do oligo pool, or we already synthesize our oligos and do like the full stack from like, we get sequences from you, we do DNA synthesis, cell-free, validation, everything in-house. And we think this sort of scaling is like other like verticalizing actually helped us a lot more to just lower both costs and latency. Ultimately, we want to get to like binding affinity results not in 2 weeks, in 2 days, and we think we can do that. Like if we like fully schedule all of our machines, fully load up everything, we can do that in 2 days. So that's another way we've been doing this.
Okay, very cool. Maybe last question, and then I will let you go. Let's assume that somebody has a protein engineering goal, like they want to design something and they have the computational resources to do that. Up to what point can Adaptyv help with the experimental assay validation? Like not all computational designers know how the wet lab works. So like how does Adaptyv help in that aspect?
Yeah, I mean, ultimately we want to be like the sort of experimental infrastructure. So we don't want like these protein designers to think, like, especially if they come from computation background, we want them to directly submit sequences, get results, and then iterate faster via us. But we also have people that have some expertise or have some knowledge, or people from pharmas, from biotechs that have sort of bespoke assay requests. And if we can already do those in our platform, and it doesn't require us like building new infrastructure or like buying new machines that are just way too costly, or like allocating too much, like too many resources into this sort of assay development. We can easily do that as well. So we're both doing like sort of, if you have like a more niche, more specific, like you want, let's say, like give an example, like binding under these conditions, or like pH responsive binders, or you add a small molecule and you want to test that, we can also do that for you. But ultimately, like the main goal, or like how we see the future more and more, especially with these agents, is that everything will be like kind of automated. You as the protein designer will have like an intent that you're showing to the model, and then everything is taken care of, and then these things are also self-improving. So then in your case, you want something that's already like an experimental infrastructure that's already streamlined, that you don't have to think too much about, or like debug too much. You want something that's robust and works in 99% of the cases.
Nice.
Hopefully that answered your question.
Definitely. Because specifically, I was thinking, okay, let's assume I know already what assays I want to run. Then
yeah,
you can do it. But also, if I don't know anything, you're still there, right?
Yeah, I mean, the easiest way, we have like this support thing. We have people asking us like, okay, I've been playing with this model, I have no idea what I'm doing, or I want like, or asking us for like custom assays. And then the easiest way for us, if it's something that's not directly available in our platform, like a simple like a BLI or an SPR assay, we discuss with that customer. We're like transparent about, okay, this will like require these resources, or like this time for the sort of R&D for this bespoke assay, and then we can also try to make that work. Like we had some customers asking us for some protease assays, and we also did that. We've been doing like some more custom stuff, especially for enzymes, like building a lot more assays that the customer had the initiative to propose that to us, and we're like, okay, we can fit that into our infrastructure. So there you go, we're doing that.
Very cool. All right. Thank you so much. It was a very interesting presentation. And yeah, for the audience, check the events that are remaining for the Protein Engineering Festival. We still have 2 more weeks to go. This was the methods week. And then we will have next week for the applications of protein design. And the final week will be at the frontiers. So new kind of designs, more than just single protein designs, etc. So stay tuned. Thanks again, Tudor. And
yeah, thank you for inviting me.
See you around.
Yeah.